In this video, we're going to ask how molecules get in and out of mitochondria. So, you do glycolysis in the cytosol, and then you do the citric acid cycle in the matrix. How in the world does stuff get in between those two? So there's our question. How do molecules get in and out of the mitochondrial matrix? So first of all, the citric acid cycle occurs in the matrix of mitochondria, whereas things like glycolysis occur out here. So glycolysis is out here. The citric acid cycle is in here, and somehow we have to be able to get across that space. Quick review of mitochondrial anatomy is inside of this, the matrix is defined by being surrounded by this, we're moving around. We're just going to stay there, I guess, by this this inner membrane that is surrounds the matrix, and then this outer membrane, which is touching the rest of the cell. And these matrices, or these membranes, differ in their permeability. So the outer membrane is fairly permeable. Most molecules can go through it. The inner membrane is not. And that really causes a problem, right? Because if you're doing glycolysis out here and you want to do the citric acid cycle in there, how do you actually get your stuff through? Okay, so when I teach this in person, I do this exercise. So this is good exercise if you want to pause and think about it. So the exercise is, here's all the stuff we have out in the inner membrane space, sort of facing the cytosol. Which of these can get into the matrix without help? So, this is a block of metabolically important molecules. Which ones can do so without help? Good chance to pause and think about it. So you can pause. All right, I'm assuming you're coming back from your pause, and the answer is not a lot. So the gases, carbon dioxide and oxygen, those can both do it. And the other ones, maybe sometimes, but not really. Maybe ketone bodies, although they actually have a transporter too. So overall, not very much can get in and out of the matrix. And this is actually really important. I haven't talked about this yet in my videos, but maybe in the past you've heard about oxidative phosphorylation and pumping protons across bilayers. This is the bilayer that does it. So it's actually really important that this be like a tight packed, super sealed membrane, a bilayer that stuff can't get through. So somehow or other, we have to solve this problem of getting a bunch of things across the bilayer into this matrix. How do we do it? And it turns out there's two answers. They're built around the idea of using transporters. And so let's talk about those, and then we'll talk about the exotic ways that mitochondria assemble these transporters to do this process. So here's an example of a transporter. Most of them are members of this family, the SLC25A1 family. I guess I can come back now. This SLC25A1 family, there's just a whole bunch of different members. We'll see in a second. They do things like the antiport of malate and citrate. So this protein will take a malate and it will move it one direction, and it will move a citrate the other direction. It is a bidirectional transporter in that it will, we've drawn it going this direction. It could also very well do the opposite direction. But when it does it, it will move them opposite each other. So it will always move a citrate and a malate in opposite directions, but they will follow a concentration gradient when they move. And this process is called an antiport because it's moving one in one direction, one in the other. And it turns out there are a whole bunch of different transporters that have distinct specificity. So let's zoom in and just look at some of them. This transporter, the GC transporter, moves glutamate by itself. Glycine moves by this transporter by itself. We've got, just randomly checking them, or looking at them here, this one is an antiporter that moves malate and phosphate. This one here is a really important one. It actually moves ATP and ADP. So basically those can swap with each other going across the membrane. But, you could do this whole survey of them and see that there are a bunch of them. And this is actually an area of active research. There are molecules that we still don't have a good understanding of how they get in and out of the mitochondria. And people think it's probably one of the bajillion transporters, but we just don't know which one yet. So currently something that's actively being studied, but hopefully you can appreciate here, even with what we know, there's already a bunch of different transporters to worry about. These transporters come in a couple of main varieties, well, three. One are uniporters, which means they just move something in a direction. The glutamate transporter we looked at here is a uniporter. There are symporters, which would move two things in the same direction. Most of these SLC25, I can never say this name, most of these SLC family transporters are not symporters, but they're a class of transporter that exists. And then actually there are antiporters. And if you look across this protein family and mitochondrial transporters, they tend to be these two classes of uniporters and antiporters. these can be assembled into machines that move stuff. And so here's a really sort of simple example. Not a simple example, but it only has 3 parts, so it's going to feel simple relative to what I'm about to throw at you. But this is a system that couples ATP synthase. This is the molecule that makes ATP in the mitochondria. Don't worry, we'll do a video about it soon. It's got the ADP-ATP carrier, and then this phosphate carrier. What I'm going to do is play this video that was generated by this group that published this paper. Come on, play. Where we're going to look at what happens, how these can work in concert. So an ADP molecule binds at the ADP-ATP carrier. That allows it to diffuse into the mitochondria. An inorganic phosphate can diffuse through the phosphate carrier. Now we've got both of these in the matrix. Once they're in the matrix, the ATP synthase can do its job, which involves allowing the flow of protons. You'll see a proton flow happening down here. Allowing the flow of protons while also doing a phosphorylation of making ATP. That ATP can then go out through the ADP-ATP carrier and then be used in the rest of the cell, right? So overall, what we're seeing here is that you can get this coupled transport activity just by moving, just by putting a bunch of these things together. Okay. But we still have a problem, and that is not every type of molecule has a transport. So everything here on the left, yeah, I'm not hiding everything. We actually have some notion there's a transporter for it. Everything on the right, there isn't. And how in the world can you do something like the citric acid cycle if you can't get NADH or acetyl-CoA in and out of a mitochondria? So I wanted to talk about one specific way, and that is the transporter for NADH. And this is something known as the malate-aspartate shuttle. I don't know, it's up there in my top 5 coolest metabolic mechanisms. So I'm excited to teach you about it. Okay, so our problem is we have NADH out here that we want to put inside of the matrix. And if we can get it inside of the matrix, we could take NADH, turn it into NAD+. We could take molecular oxygen and make water. So we do this favorable reaction that we're going to actually couple to ATP generation. So this is what we want to do. We're stuck out here. How do we do that? And this is kind of like one of those 2048, the game where you're trying to like line things up in particular patterns to get things forward here. So we're going to do a set of moves that when you work it all out, transport happens, even though transport doesn't even seem to happen. So let's try it out. Okay, so first thing we're going to do is we're actually going to take NADH and we are going to oxidize it to form malate. Okay, so this is actually the reverse of a reaction we saw in the citric acid cycle. This is a thermodynamically favorable reaction actually to go from oxaloacetate to malate. We are doing a reduction of that. So now our electrons are on the malate. We're then going to, on the opposite side of this, we're going to take a malate and we're going to take the electrons off. We're going to put it onto an NADH and we're going to ultimately recycle that using our oxygen. So this is a very favorable reaction on this side. But what in the world? We did something out here, we did something in here. How does that couple? Well, now we're going to add one of these transporters. And what this transporter is going to do is it is going to move the malate inside so that this can happen, right? So now we've taken an oxaloacetate, we've taken the electrons from the NADH, stuck them on the malate, take the malate in, drop them off here. So we've net taken electrons, right? They were here, and we've stuck them here, right? We stuck them onto oxaloacetate, it carried them in, and it dropped them off, and now they've gotten, so we've achieved what we wanted to do. We've moved the electrons inside the cell. But what in the world's going on here? We also have this alpha-ketoglutarate transport going on, and we also move the oxaloacetate in. I don't know if we want to move an oxaloacetate, and this is not a sustainable reaction. So what do we do next? Well, we add more stuff to transport. So now we're going to add another transport activity. And in this transport activity, what we're going to do is we're going to move a glutamate in and an aspartate out. And then we are going to do some conversions. We're going to do what's called a transamination reaction, where we take a glutamate, we're going to take the amide off of it and stick it onto oxaloacetate. That will generate an aspartate. The glutamate is converted into an alpha-ketoglutarate. So now we've sort of completed the inner side of this loop, right? And we've made an oxaloacetate and an aspartate here. We then complete that outside. Now we're taking an aspartate Yeah, I'm still not hiding things. The aspartate gets turned into an oxaloacetate inside, and the amide gets transferred onto the alpha-ketoglutarate, onto the glutamate, right? So what have we done here? Let's think about the net reaction. So an oxaloacetate goes in, goes through this whole cycle, comes back as an oxaloacetate. So we haven't consumed any oxaloacetate or made any. The malate gets made and then goes away, right? So nothing's happening to it. Malate goes here, goes in a loop, comes back to being malate. Same with the aspartate, right? You made an aspartate here, but then you go around and you make another aspartate. Inner cycle, it applies. Glutamic acid, glutamate goes to glutamate, alpha-ketoglutarate, alpha-ketoglutarate, glutamate. So that's an inner cycle. With the amines, right? So that we have an amine that gets transferred and goes in here, but then it gets transferred and goes out here. Every cycle that you make is perfectly conserved across it with the exception of the electrons and actually this proton. One proton is transported across. This is the aspartate malate shuttle. It has this net reaction of taking NADH in the matrix, or in the IMS, and an NAD plus in the matrix. It then makes an NAD plus in the outside and an NADH in the matrix, along with a proton that gets transported. It's a very cool mechanism. And what's driving this mechanism forward is from that oxidase of NADH, right? This thing right here, Le Chatelier's principle, it's always going down, right? It's being burned with oxygen. So because of that, you have this net drive for this reaction. Even though it's all this complicated indirect shuttling mechanism, you can still move electrons all the way through the system being driven by this oxidation reaction. So I'm going to go back and say it again. Another way to think about this is we're taking our electrons and we're making them a passenger on the malate. And then we're taking the malate and it's dropping off the electrons here. So it moved them as a shuttle. And then all the rest of this complicated stuff is just mass balancing energy, mass balancing the system so the shuttle basically can go home. You can think about it as a delivery truck that it drops off. Come on, pen. There we go. Drops off its electrons, and then this is its return route home, maybe refueling, whatever helps you think about it, so it can pick up electrons and do the loop one more time. Okay. And this is actually a really common mechanism for getting things in and out of mitochondria and even like through the bloodstream, where you'll know we'll come across these. So we've got here, for example, this is the carnitine shuttle, where carnitine on one side, which is a molecule involved in fat metabolism, you'll notice there's carnitine on both sides, but the carnitine itself is crossing to move in because an acyl can't. An acyl CoA, a CoA is on both sides, right? So CoA to carnitine, carnitine to CoA. We're moving CoAs without moving CoAs. Okay, I don't want to go into details on that because we've already done a whole bunch of details on the malate-aspartate shuttle. So zoom out again. How do molecules get in and out of the mitochondrial matrix? There's direct transport. There's these SLC25A carriers. There's a bunch of them. They can be uniport. They can be antiport, for example. And then there's also this indirect mechanism, which basically you couple these shuttles with concentration gradients in Le Chatelier's principle to take moieties of interest through the system that are attached to carriers. So, for example, electrons that get stuck onto a malate and moved inside of a mitochondria is an indirect way to take an NADH from outside and make an NADH inside. Okay, so hopefully that helps you now as you're thinking about metabolism and things like how do you get into the citric acid cycle. I look forward to seeing you in future videos, and thanks for taking the time to watch this one.